We're going to review a variety of biology lab things today. We're going to talk about enzymes, cellular respiration, photosynthesis, mitosis, meiosis, and DNA biology and technology. So this is a really broad swath of information today that we are covering. All the chemical reactions that occur in a cell, these are collectively called what? This is called metabolism. If the things that are the beginning parts of a chemical reaction are reactants, what do we call the things that we end with? They're called the products. Thank you. These are organic catalysts that speed up metabolic reactions. Enzymes. They're enzymes. And I think my most famous one is carbonic anhydrase, and I've looked this up on the Wikipedia, and I know that nobody that is legitimate likes to think Wikipedia says anything true. But I have read on Wikipedia that carbonic anhydrase, one molecule can catalyze anywhere from 10 to the 4th to 10 to the 6th reactions per second. That's 10,000 to 1,000,000. Unbelievable that one molecule can do that. And this is an important enzyme with the conversion of carbon dioxide back and forth in your blood and respiratory, so that the waste product from your body can be eliminated from your body through breathing. Here is a peculiar science word. The things, the chemicals that participate in a chemical reaction are called what? They're called substrates. And your book actually says they are the reactants in the enzymatic chemical reaction. What do we call the sweet spot on an enzyme where this substrate engages?
Active
It's
site.
called the active site. And if I wanted to denature an enzyme or denature a protein, would that be a good thing or a bad thing typically? Does that help the enzyme or hurt it? It hurts it. It says it alters its normal shape in a way that inactivates it. By the way, my favorite denatured protein is egg white, probably the yummiest one of all. I do like them fried over medium, possibly scrambled with cheese on top, and I'd like it in an omelet. So I actually do like denatured proteins myself. Sometimes I eat them with bacon. Sometimes I eat them with green eggs. Sam, I am. All right, let's skip over to cellular respiration next. Aerobic respiration is where we knock out our bodies, we can say our bodies are good at this. 38 ATPs per molecule of glucose with aerobic breakdown. And when I say aerobic, what does that mean, class? It uses oxygen to make this happen. So if we have anaerobic fermentation or breakdown, this can actually do several things. If it's a yeast, it can wind up making food products like breads or beers or other yummy things like pickles. I don't know if it's a yeast that does that, but it sure has a nice tart flavor to it. In humans, if we try to participate in an activity that is too much for us, we get sore. So what kind of fermentation is that called?
Lactic.
It's called lactic acid fermentation. You only get about 2 ATPs per molecule of glucose in all of the fermentation processes. The good news is that the chemicals that participate in the making of the lactic, let's say this, the lactic acid will eventually be broken down into its component parts. You don't stay sore forever. So this process takes glucose and oxygen and breaks it down into carbon dioxide, water, and 36 to 38 ATPs. What am I talking about? Talking about straight up aerobic cellular respiration. Nice. Let's talk about photosynthesis just for a few minutes. Where does photosynthesis take place in a plant cell? It takes place in the chloroplasts. There are little discs called thylakoid discs, and they stack up to make these certain things. What are they called? They're called grana. A singular stack is called a granum. And it's during the light reactions that ATP and NADPH are formed. So if it's the light reactions that form those things, what does the Calvin cycle produce? It produces at least the precursors to sugar. And we learned in our regular book that it's glyceraldehyde 3-phosphate, it's G3P. In here, it just calls it a CH2O fragment. That's what your book calls it. If the first part of photosynthesis occurs in the stacks of thylakoids, where does the Calvin cycle occur? It occurs in the stroma, or the liquid part of the chloroplast. And another thing that we learned in class, in regular lecture class today, is that there are things called stoma, or holes in plant cells, that allow carbon dioxide to come in and oxygen to go out. But here we're talking about the word stroma, not stoma. In a graph, what wavelengths of light do plants like best? Is it green and yellow or red and blue? It is red and blue. Plants actually reflect like green and yellow. Let's talk about mitosis for a little bit. Actually, this is before mitosis. What do we call the phase where the cell is performing its usual cellular functions? Interphase. What is the part that holds chromatids together in the middle? Centromere. Okay, now listen close to this, class. Centromere holds chromatids together. Centrosome is where the centrioles are found. So we got a centromere, a centrosome, and centrioles. That is kind of confusing to me. A lot of words with C and TR in the middle. If mitosis is the division of the nucleus, what is the name for the division of the cytoplasm in itself? Cytokinesis. Cyto means cell, kinesis means movement. Okay. What would you call the division of sex cells? Excuse me. This is called meiosis. Okay. And remember, just kind of conceptualize for just a minute. Why is it important for a 46 oogonium or spermatogonium to go down to a 23 egg and a 23 sperm? Why is that important? Say it again. So that they can combine and make 46, make the full human count, right? So we have two haploid gametes that combine to form one diploid zygote. Very good. When we look at the structures associated with mitosis, this is a large organelle containing the chromosomes and acting as a control center for the cells. What is this called? It is the nucleus. What do you call the microtubule structure that brings about chromosome movement during cell division? It's called the spindle. This is a constriction where duplicates or sister chromatids of a chromosome are held together. Centromere, okay? This is the central microtubule organizing center of the cells. It contains two centrioles, by the way. What am I talking about? Centrosome, and then these are short cylindrical organelles and animal-cells that contain microtubules and are associated with the formation of the spindle during cell division. What am I talking about here? Now we're talking about centrioles. What do we call the short radiating fibers produced by the centrioles? It is called the aster. And listen to this, class. In science, aster means star. Have you ever heard of the word asteroid? Mm-hmm. It's like something that is thought to be a star. I noticed that in Guardians of the Galaxy, that guy that has that big hammer, he's kind of like a bad guy, and I can't think of his name. Ronin. Okay, thank you. It's not Thanos. Thanos is the guy that looks like a bullfrog, right? Okay, so it's Ronin. His spaceship, I think, is called the Dark Aster in that movie. So the Dark Star. Dark Star. But he does have a really cool dance-off with who? Chris Pratt, Peter Quill. Okay, I'm sorry. I couldn't think of the movie name. I could only think of the pretty cool dance-off. We need to watch that. That's good living right there, by the way. That is true life. All right, let's keep on going here. This is the nuclear division that results in 2 new nuclei, each having the same number of chromosomes as the original nucleus. This is called mitosis. If the parent cell is the original cell, what do we call the 2 cells that result? These are called daughter cells. Thank you. When we look at a chromosome, what do we call the little grabby places on either side of the chromosome where the spindle fibers connect? It's at the centromere, but we're actually talking about little places called the kinetochore. Kineto means movement, choreograph means dance, so these are the grabby sites that allow the chromosomes to dance across the cell as it divides. What phase in mitosis do we call it when the chromosomes line up at the very middle? Meta, metal, brita, meta. Yes, metaphase. What do we call it when the chromosomes divide in the middle and the chromatids each go to different poles? It's called anaphase. What do we call the phase when the daughter cells form? It's called telophase. Okay. In an animal cell, we say that the division is called a cleavage furrow, but in a plant cell, we call it something else. And it's on the next page. It's called a cell plate. In metaphase, there are homologous chromosomes. Excuse me, let me rephrase that. In metaphase of meiosis 1, there are homologous chromosomes that line up next to each other, and one tip of this one switches with the other tip of that one, and it provides genetic diversity. So if we call the part where they line up together synapsis, what do we call when they actually exchange the genetic information? It's called crossing over. In meiosis, if we start with one cell, how many do we get in the end? If we start with one, how many do we get in the end? You start with one spermatogonium, and we get how many? Does my book has 1, 2, 3, 4. Now, if you're a female, you don't get 4 eggs. You actually get 1 oocyte and several polar bodies that help just divide the DNA. So it's kind of different for the ladies. We knew the ladies were special. Now to the ladies. Now to the ladies. Yeah. Okay, let's talk about some things that can happen to chromosomes that are not actually good. What happens if you get an extra chromosome on the 21st pair? This is called Down syndrome, and it is actually called a non-disjunction. Tell me what this is. The chromosome is shorter than usual because some portion is missing. This is called a deletion. The chromosome is longer than usual because some portion is present twice over. It's called a duplication. The chromosome is normal in length, but some portion runs in the opposite direction. Inversion, and a chromosome completely switches places. What do you call that? Translocation. That's right. All right, remember that in meiosis, how many sperms do we get from one spermatogonium? We get 4, but how many oocytes, how many functional eggs do we get? We just get 1. And what do we call the little tiny parts of the lady division that just allows for DNA to divide? It's called polar bodies. And remember that all of the cells resulting from meiosis are diploid, right? They have the full count. That means sperms have 46, eggs have 46. No, that is false. They are haploid. Remember that. We got 23 added to 23 to make 46. If the seminiferous tubules happen to be the site of sperm production, where is the testosterone produced? Everybody say interstitial cells. Interstitial cells. Okay, now let's skip over to, let's talk about DNA for just a minute. DNA is the code of your body, really. And RNA is what manipulates the code. It copies it, makes proteins from it, regulates it. Because the RNA, listen to me, because the RNA is so important in the function of the cell, evolutionists actually believe that RNA came first, because if there was a code but nothing to read it, manipulate it, interact with it, then what's the point? So that's called the RNA first hypothesis. This is when a DNA makes a copy of itself. What am I talking about? It's called replication. In RNA, there is one base that is different than in DNA. Remember, A bonds with T, C bonds with G, those rungs of the ladder in the spiral staircase. What's the difference in RNA? There's a purple one in your book. It's purple colored, and it is uracil instead of thymine. So what do we call the phosphate, sugar, and bases that repeat? There's a unit. It's called a certain unit. It's called a nucleotide. This is the repeating unit that we see in RNA and DNA. What are the three kinds of RNA? Messenger, ribosomal, and transfer. So messenger RNA is what copies the DNA. Ribosomal is where the protein is made. It's called translation. And the transfer RNA is what shuttles amino acids over to the ribosome for assembly. And remember, listen to me, there is a codon on the RNA, messenger RNA, and then there's something that matches to it on the transfer RNA, and it's kind of equal and opposite. If this is the codon, what do we call the part that fits on the transfer RNA? It's called the anticodon. Everybody say anticodon.
Anticodon.
What do we call it when the RNA copies the DNA? It is called transcription. All right. What do we call it when the DNA is made from that code? It is called translation. Let's go to that chart and look at that chart just for a few seconds. We won't be there very long. There is one amino acid that's always the first amino acid in a protein chain. What is it? Everybody say methionine.
Methionine.
And the codon is AUG for that. What do we call UAA, UGA, UAG? Why are these special? These are stop codons, and these tell the ribosome to stop reading the messenger RNA, to disassemble. It's a signal for the protein to be completed. And what's bizarre is even when a protein is completed, it has to detach and fold upon itself in a magical way before it can be functional. So that's one of the things that we're kind of learning in genetics and DNA, is that just knowing the code is really not enough. There's more magic to it than that. Pretty interesting. So it kind of, in a way, if you really think about it, it kind of makes you think that even DNA, listen closely, this is kind of it kind of gives you the idea that even DNA is more than the sum of its parts. That's kind of scary for some people because that doesn't sound like science. But there's something else going on that helps regulate some knowledge or something that I don't think scientists have quite figured that out yet. If you watch Star Wars, it would be called like a life force, like a midichlorian count, something like that. And in chiropractic school, they called it innate. They said there is an innate life force in living things that gives them. We might want to call it spirit or something like that. That would be another word, wouldn't it? Some kind of life force. So, to me, that's one of the dilemmas of being a scientist, is accepting that very thing. Are you really only the sum of your parts, or are you really more than how you appear? I happen to think that second thing. I
Me
think
too.
I think you're a little bit more than a sum of your own components. All right, so we got our review done. Marvelous. Very good. Thank you, Mary.